Hey everyone, welcome to another episode of the podcast, episode 62. Today I'll be discussing fungal hydration and fungal nutrition. Basically, how fungi get the nutrients that they need to stay alive and to grow. This is going to be a really interesting episode because, unlike plants and animals, fungi have a relatively diverse range of methods that they use to acquire their nutrients. Plants will typically absorb nutrients out of the soil, and these get carried through their vascular system to the places where growth is actively occurring, like meristems, leaves, and the vascular cambium that generates secondary growth. Now, animals get their nutrients from consuming other organisms, be they plants or other animals. And the consumed organism gets dissolved into its base nutrients within a series of internal organs, like stomachs and intestines. So that's how animals do it. Now, fungi have a variety of mechanisms that various species use to acquire their nutrients. Some fungi are parasitic, and they eat from or they steal nutrients from their host, even in cases where the host is still alive. Some fungi engage in symbiotic relationships with plants or algae, and they collect food through a cooperative optimization strategy. Most fungi are saprophytes, or saprotrophic organisms, and this is a really cool way of digesting food that I'll explore in detail in a few minutes. But first, I want to start by discussing fungi hydration. Relative to almost every other aspect of fungal physiology and life strategies, their hydration is probably the simplest and most straightforward thing about them. In general, the hyphae of the mycelium have a huge surface area relative to their volume, and this makes them hugely absorptive of the water and free nutrients that exist in the soil. When the hyphae grow through the soil, they'll simply absorb water through diffusion, and the water will be integrated into the cytoplasm that flows through the hyphal network. This absorptive quality is what can make fungus as much as 90% water by mass. But there's a downside. Because of their extremely high surface area, water doesn't just get absorbed easily, water also evaporates out of the fungus really easily. It evaporates at a really high rate. This confines fungus to humid areas, in the shade, in the soil, away from excessive light and heat that would be lethally dehydrating. However, fungal hydration isn't as simple as just diffusion and evaporation, if only. In reality, in many cases, fungi will create specialized hyphal structures, and these are kind of optimized for water absorption and water transport. And in that sense, they're a lot like the roots of vascular plants. These structures are called mycelial cords, or rhizomorphs, which is translated literally to root form. These rhizomorphs have structural qualities that mimic the vascular tissue of plants, and it allows them to transport water and nutrients across relatively large distances. This compensates for the fact that diffusion can only move water a relatively short distance, and it's unable to keep a large fungus totally hydrated. These mycelial cords are composed of bundles of hyphae, all running parallel to one another, creating a thick rope of hyphae filaments. This is why the mycelial cords look relatively large. They look thick, like plant roots, whereas when you just have an individual hyphae, it's typically microscopically thin. The hyphae on the inside of these rhizomorphs are typically wider. They have a larger diameter, and they're called vessel hyphae. And these are surrounded by a coating of smaller, narrower sheathing hyphae. They together form a sheath. Now, when you have both these sheathing hyphae and these vessel hyphae, they create a relatively conductive corridor that can carry the water and the nutrients across a long distance, and they can thus be used to feed a fungus that has its hyphae spread across a wide area. This provides a valuable evolutionary function to the fungi, as it allows them to situate themselves on a primary food source, and then it allows them to support the growth and expansion that's necessary for them to reach out and explore their local habitat to find more food sources. As the mycelial wavefront expands and grows farther and farther away from the primary food source, these rhizomorphs can support continual growth by piping in water and nutrients from as much as 9 meters or 30 feet away. In fungi that parasitize a host, the rhizomorphs allow the fungi to spread through the body to establish new mycelial wavefronts, new zones of infection in new areas of the host's body. Pathogenic fungus have a variety of chemical mechanisms that they use for acquiring nutrients, and each mechanism has been shaped by evolution to optimize for the parasitism of a specific host. Sometimes there's a species that can be infected by more than one kind of fungal parasite, and each fungi species has its own mechanism for infecting and stealing nutrients from its host. For example, there are no less than 6 fungi species that can parasitize and infect humans, and they each have specialized means for infecting us and doing particular types of damage to our bodies and spreading through our bodies. For example, there's a species of yeast fungus typically found in bird feces that's called Cryptococcus neoformans. This species can infect a variety of plants and animals, including humans, where it will typically infect the lungs. Here's what happens in the course of this infection. The spores get inhaled somehow, most likely through proximity to bird feces, and they get inhaled into the lungs, where they come into contact with the macrophages of the lung's immune system. These will try to fight off the fungal invaders with oxidative and nitrosative chemicals, and this will kill some of them, but not all of them. This will actually encourage the few surviving C. neoformin cells to get eaten by the macrophages, but before this happens, they produce a protective capsule coating, and so when the macrophages come by and eat them, instead of getting dissolved and digested and broken down, they actually survive within the immune cells themselves, and they lay dormant. From this position within the lungs, they can easily be spread into the blood, and then pass through the blood-brain barrier of the central nervous system, where they can infect the brain and the spinal cord. Here, the C. neoformans feeds off of the glucose and the ion nutrients in the blood and the brain, and they can cause serious diseases like meningoencephalitis, and brain abscesses called cryptococomas. Basically, by infecting the bloodstream, especially the nutrient-rich blood in the brain, the fungi can easily absorb everything it needs to survive right out of the human host. It absorbs its food directly out of our bloodstream, parasitizing nutrients in what is the most intimate way possible. Many notoriously brutal parasitic fungi species come from the Ascomycete genus Cordyceps. These typically infect arthropods like crabs, prawns, and insects. These fungi have a horrible and violent means of extracting nutrients from their host. Their spores get consumed or absorbed by the insect, or the hyphaea of a pre-existing fungal individual will latch onto the exoskeleton and penetrate it, reaching into the soft guts inside. Once it's penetrated the exoskeleton of the arthropod, the mycelium grows deep inside the host's body. As it grows, it literally feeds off of the internal tissues of its host, eating its soft guts from the inside out. Some of these species, like the Ophiocordyceps unilateralis, induce a zombie state in their hosts. They do this by hijacking their bodies, changing their behavior, even controlling their gene expression, and this allows the parasitic fungus to steer around the insect's body like a flesh vehicle. The fungus steers the insect's body to a place and into a position that's advantageous for the spread of the fungus's spores. These spores then go on to infect more insects, and the fungus spreads. I'll talk about this in much more gruesome, visceral detail in the upcoming episode on fungal reproduction, as this mechanism of infection is ultimately used to help the fungus reproduce and spread its spores. The insect host itself provides the gooey organic food for the fungus to eat, and its body, before the fungus eats too much of it up, its body becomes a literal biological vehicle for the fungus to control and manipulate at its leisure. The Armillaria genus of honey fungus are known for their parasitism of trees, which leads to widespread tree death and the destruction of entire forests. The Armillaria fungi use their rhizomorphs to come into contact with and then penetrate the roots of trees. They typically go for smaller, younger roots because they're relatively soft, or they go for roots near the trunk of the tree where the fungi can access the full load of nutrients being absorbed from the soil with relatively little investment. Once the rhizomorphs are in the tree, in the plant tissue, they grow in and under the bark, followed by the spread of white mycelia, which then begins to interfere with the vascular system of the host tree. It begins to steal its nutrients. Sometimes the armillaria will kill the tree quickly. Other times, typically with larger, older trees that have huge, expansive root systems, it'll take a little bit longer for the host to die. If the infection progresses over the course of years, which is entirely possible, the tree will experience symptoms of degrading health, like progressively worse and worse leaf growth during every summer, every growing season, because the fungi infection is progressing and stealing more and more of the tree's nutrients. Eventually, after having suffered from infection and relative starvation for months or for years, the tree will eventually die. Now, most parasites, like insect parasites in an animal, will also die if their host dies, so it's in their interest to moderate their parasitism so that they can get the nutrients they need, but also so that they don't outright kill their host, because then they won't be able to feed off it anymore if it's dead. They won't be able to enjoy its metabolic byproducts if there's no life anymore creating those metabolic byproducts. This moderation of their own infection is an issue of life or death for most parasites, and so they typically want to avoid committing suicide by proxy. However, fungi like armillaria, they just don't care whether their host tree lives or dies. It doesn't matter to them. In fact, it actually probably prefers it if the tree dies, because the fungi can still consume the dead tree matter through saprophytic metabolism. Now, saprophytic nutrition, or saprophytic metabolism, is, in my opinion, one of the cooler ways that organisms on Earth have evolved for getting their nutrients. Saprophytic or saprotrophic organisms are those that engage in the extracellular digestion, or the external digestion of their food. Typically, the saprophytic organism will find dead and decaying organic material, and after growing on it and permeating it, the hyphaea will release digestive chemicals that begin to break down the detritus and turn it into a soft, chemical mush, which is then really easy for the fungus to absorb. In some cases, an organism may be stressed by a harsh environment or by some random disease or injury. Maybe these factors combine to kind of put it into a terminal decline, or maybe the plant is it's at an old age, it's dying down. It's not dead yet, but it is dying. In this case, fungi engage in what's called facultative saprotrophism, where they infect the dying organism and speed up the process towards its death. They infect the host, and then they just begin eating it right away. They begin secreting these digestive chemicals immediately. And it literally dissolves the host. And because the host isn't dead yet, it has to suffer through literally being digested alive by this fungus, until it does eventually, from all of the stresses compiling on it, until it just outright dies. When that finally happens, when the host finally dies, the fungus is no longer slowed down or hampered by the host's immune system, and so the saprophytic digestion really begins to take off. The armillaria honey fungus that I just talked about is one such facultative saprophyte. It infects trees through their roots, typically trees that are already stressed or in the process of dying, and it pushes them along with compounding infections and parasitism until the tree dies. Then, the tree's entire body becomes fair game for the fungus to consume. The fungus can take years to fully digest a dead adult tree, which yields an absolutely tremendous amount of nutrients. As the armillaria, or any other facultative saprophytic fungus for that matter, digests its most recent kill, it now has a home base of nutrients, like a central source of nutrients that will allow it to spread out and explore more area for more hosts to infect and kill, or for more already dead detritus that it can consume. The nutrients from the rotting tree will support the fungus's growth as it sets up lethal attacks on other trees, and in this way, it has the potential to go from tree to tree and wipe out entire forests in a seemingly unstoppable wave of mycelial expansion. Okay, so let me explore this technique of saprophytic digestion in greater detail. First, there has to be a specific condition, or set of conditions, for a saprophyte to be able to effectively break down detritus and then absorb the product nutrients. As I explained earlier, the fungus will need a steady supply of water. The fungus is overwhelmingly composed of water by mass, and so water is necessary to keep it healthy enough to engage in metabolic activity. Water is also a necessary ingredient in some of the chemical reactions involved in digestion, so without water, those reactions wouldn't be able to take place. It's kind of like how photosynthesis requires water, which gets split into hydrogen ions and molecular oxygen. Without water, a plant can't conduct photosynthesis, and its carbon synthesis grinds to a halt. The same goes for fungi and their carbon-acquiring equivalent. Furthermore, the digestive enzymes secreted by the fungus require an external aqueous solution in order for them to dissolve and disperse across the substrate. The substrate being the detritus, the dead or the dying organic matter that the fungus is trying to consume. They need a water solution or some other suitable solvent so that they can float around their digestive enzymes so that they can get into contact with and break down as much of the biological substrate as possible. This is all about surface area. Without this watery solution, without this liquid matrix that helps the digestive enzymes just cover everything, they won't get very far. The digestive enzymes might end up just coating the fungus itself, which could be counterproductive and potentially damaging. Second, the temperature has to be suitable for fungal metabolism. If it's too cold, the fungus will be unable to effectively engage in the metabolic processes necessary to break down detritus. The energy balance will be all out of whack, and the requisite chemical reactions just won't work. The coldest temperature that saprophytic fungi can tolerate is around 1 degree Celsius, or 33 degrees Fahrenheit, which is just barely above the freezing point of water. If it's too hot, on the other hand, the necessary enzymes will also be unable to function. Perhaps they can't become activated at high temperatures, or perhaps they denature and they become useless. Whatever the case may be, saprophytic digestion typically doesn't work at temperatures above 35 degrees Celsius, or 95 degrees Fahrenheit. Studies have shown that the optimal temperature for saprophytic fungi to absorb nutrients, and to use those nutrients to grow and to heal themselves, is on the warmer end of this range, so around 25 degrees Celsius, or 77 degrees Fahrenheit, which is about as warm as a typical summer day in the mid-range and higher latitudes. Furthermore, to optimally engage in saprophytic digestion, the fungus needs to exist in soil that's either acidically neutral or very slightly acidic. So the soils that you'd find in a boreal forest or a peat bog in the northern taiga will tend to be, in general, much more acidic than your average saprophytic fungi can handle. Lastly, saprophytic digestion is an aerobic process, which means it requires oxygen. The vast majority of saprophytic fungus find it very challenging to digest detritus in anaerobic conditions, like you would find in wet clay soil, or a swamp saturated with water, or really in any other kind of water body like a lake or a river. Sometimes, even dry soil doesn't have enough oxygen to support saprotrophism, so the fungus is restricted to detritus that's littering the ground. Now, I say restricted, but this really isn't much of a restriction, because when any kind of plant or animal dies, its body almost always ends up just resting on the surface of the soil, where it's exposed to the air, and all of the scavengers and saprophytes that want to come and take a chunk out of it. So these are all the conditions that are necessary for the fungus to break down detritus into bioavailable food. The next topic is then how the specific biochemicals in the detritus get broken down and metabolized. Just like plants and animals, fungi require proteins, nucleic acids, lipids, and carbohydrates. However, many of these molecules are far too large to simply be absorbed directly through the hyphaea membranes. So first, the fungus has to break them down into smaller chunks or pieces before it can absorb them. In a nutshell, this is why fungus have to engage in this extracellular digestion. Just physiologically, they can't absorb stuff unless it's already digested down to its little molecular components. Once they absorb these little molecular component bits of their detritus, they then utilize their own enzymes and their own internal biochemistries to reconstitute these chemicals into the fully formed proteins, nucleic acids, lipids, and carbohydrates that they need to maintain their tissues and grow. So where you, as an animal, will eat your food, and you'll chew it up in your mouth, and you'll swallow it, and it'll be chemically dissolved in your stomach, and then it'll go into this huge, long, curvy tube of your intestine, which has it's very high surface area, because its purpose is to absorb all of these nutrients. This is happening inside of you, and so compare this to the fungus, where they can't absorb all of these nutrients as they are. They don't have a mouth to put food into, so they exude these digestive chemicals on the outside of their bodies. And what you do on the inside of your stomach, they do outside all around them, in the detritus that they're growing in. And they turn it all to mush that's really easy for them to absorb. The metabolic enzymes themselves that the fungus uses to break down its food, those are expressed in groups, where the whole group of enzymes is necessary to fully break down and absorb a particular compound, like a lipid or a protein. Because the excreted enzymatic fluid is a thorough mix of all of these necessary enzymes, they can all participate to fully metabolize a given compound. As I'm going through this, keep in mind the high surface area of the mycelia, due to all of the little thin hyphaea creeping into the substrate. This high surface area is how fungus are so effective at absorbing all of the nutrients that they dissolve out of the substrate. To my surprise and frustration, the information on how fungal enzymes break down various macromolecules was kind of challenging to find. A lot of the in-depth chemical details are just not well understood. There have been little to no studies that have peered so deeply at the specific metabolic processes. But with that said, it is known that cellulases break apart carbohydrates like cellulose. It's known that peroxidases degrade lignin, and that pectinases are used to degrade and decay the softer tissues in plants like fruits. These are the kinds of enzymes that are evolved in your typical plant digestion, as the fungus has to be able to break apart plant polymers. Now, dead animals are also a rich source of food for fungus, especially proteins. However, because the animals lack cell walls, their bodies decay relatively rapidly. As far as the fungus is concerned, an animal's carcass is a tasty but an ephemeral treat. To break apart the fats, the lipids in the animal's body, the fungi secretes enzymes that are called lipases. These lipases consume a water molecule through a hydrolysis reaction that catalyzes the breakdown of lipids. They do this by snipping a specific region of the lipid, like breaking up a triglyceride into monoglycerides, or by breaking open the aromatic rings in a steroid to crack it apart into smaller, more manageable pieces. To break down the proteins that are so heavily concentrated in animal tissues, fungi express enzymes called proteases. These also consume a water molecule in a hydrolysis reaction, using it to break apart the peptide bonds between amino acids. These peptide bonds hold the amino acids together in a chain, in a complete protein. So if you break them, you cut up the protein into smaller chunks. Although keep in mind that there is an impossibly huge variety of proteins. Despite this, these proteases are clever. The chemists call them promiscuous, but all that means is that these little enzymes have one or some main function, and their promiscuity means that they can also engage in a wide range of side reactions. Now, these side reactions are usually slower and less important. In a living organism, the side reactions are a lot less vital, because the protein isn't specifically designed to do that particular function. It has its own main function, and whatever the secondary function happens to be, it can just tangentially support those reactions. This doesn't rule out, maybe a living organism having another protein that is specifically focused on that secondary task, but that protein might have its own secondary effects. And you can just go on and on and on with all of these protein enzyme cascades. Basically, in living organisms, the secondary functions of some random enzyme, it might be nice, but it's much less important to the living organism than the enzyme's main function, whatever that happens to be. However, this is not the case for the fungus. To the fungus, what this protease promiscuity means is that digestive enzymes are not just doing their main function, which is to break down the proteins, they're also doing a lot of secondary reactions. And these are all like little bits of extra work breaking stuff up. This is good for the fungus, because all of that enzymatic promiscuity, really, it adds up, and it really improves the rate of their metabolic, I shouldn't say digestion quite yet, their metabolic dissolving of the substrate. When you express millions of these enzymes, all their little side reactions will accumulate into a non-trivial portion of the total digestive activity. It ultimately helps fungus speed up the breakdown of their food, and this yields more nutrients per unit time. That has an obvious advantage for the fungus when the animal carcass will largely be scavenged to the bone within a couple of weeks. Perhaps the most common carbohydrate that a fungus runs into is cellulose. Cellulose is by far and away the most abundant biopolymer on Earth, as it's present in the cells of literally every plant and most algae. Cellulose is a biopolymer. It's composed of monomers of glucose. To first break down the cellulose polymer, the fungus will express an agent called a cellulase. These cellulases will go in and consume a water molecule in a hydrolysis reaction to break apart some point in the polymer, or I should say, some specific type of chemical bond within the polymer. The water molecule gets split apart, and it's used to cap the ends of the smaller polymers that are created by this split. For the cellulose to be fully broken apart, it requires several different kinds of cellulases, each targeting a specific bond or configuration within the polymer. For example, the fungus Trichoderma resi expresses no less than 7 types of cellulases. One of them cuts up the cellulose polymer into small 2-monomer chunks called dimers, or more specific to cellulose, it's called cellubiose. These cellubiose chunks are a lot more absorbable, they're tinier so they're a lot easier to manipulate. It's easier for more enzymes to come by and attach to them to break them apart into the individual monomers. All of these things, they just have steps to play in this big, giant orchestra of detritus breakdown. When you have all of these different cellulases all working together to digest some plant detritus, the total activity of all of these expressed digestive enzymes will break down the cellulose into its constituent monomers, into many little molecules of glucose. For the fungus, the raw glucose is easy to absorb, and it's easy to metabolize to extract the chemical energy. Now, plants have cellulose, but some plants also have a chemical called lignin. Lignin is involved in the evolution of wood. Wood is one of the most abundant types of biopolymers on the planet, and the lignin within it is the second most abundant polymer. It's second only to cellulose. The vast forests that carpet much of the terrestrial landscape are covered in billions and billions of tons of wood. Wood is dense and it's heavy, it's packed with carbon and other key nutrients, and because of the cellulose cell walls and the lignin that gives wood its strength, every cubic meter of wood contains a relatively high amount of chemical energy. All of this chemical energy comes from all of these carbon-carbon bonds and all of the lignin and the cellulose. Now, if we look at cellulose, it's a polymer that's just made up of glucose monomers. If we look at lignin, we'll see that it's a polymer composed of 3 different kinds of monomers, each linking together in a random order. It doesn't require a specific order like a DNA polymer does. Specifically, there's the paracumarol alcohol with 2 alcohol groups and an aromatic ring. There's the coniferol alcohol with a similar base structure, but it also has a single methoxy group on it. And then there's synapol alcohol. which again has the same base structure, but it has two methoxy groups. Paracumaryl alcohol is typical of lignin in grass species, species of grass, while lignin in Norway spruce trees is apparently close to 100% coniferal alcohol. So the lignin in those spruce trees is just monomers of coniferal alcohol, repeating for however long the polymer is. Anyway, when lignin was first evolved sometime in the early Carboniferous period, there were no organisms that were around to digest it. No species had the enzymes necessary to take it apart and capture that chemical energy for itself. That was until bacteria and fungi evolved the means to do it, in the range of some 300 million years ago. Fungi evolved a group of peroxidase enzymes as the primary decomposers of lignin, with support enzymes called lacases that further break down the resulting chunks, or the smaller lignin polymers and monomers. Other fungal enzymes help produce H2O2, or hydrogen peroxide, which itself is necessary for the function of the peroxidase enzymes. For example, the heme peroxidases have an iron atom that reacts with hydrogen peroxide to produce an intermediary chemical that will ultimately oxidize the lignin. Another example in species of white rot fungus is that they release a chemical called veratryl alcohol, which is an upregulating metabolite that helps lignin peroxidase break down lignin. Now, I could go into a lot of technical detail here, but it would be an exercise in pronouncing chemical names, and it would get boring really quickly. So rather than doing that, let me try to express how crazy important this evolved ability of fungus really is. The capacity to break down lignin had literally global consequences. Okay, so try and take this little next sentence and put it in the paragraph that I just finished. Believe it or not, the lignin isn't actually consumed by the fungus for energy. The lignin is just getting in the way of all the cellulose, and the cellulose is what the fungus can eat for actual chemical energy, for nutrition. And so to get the lignin out of the way, fungus have evolved these peroxidase enzymes, and what they basically do is just come in like a wrecking ball, and then they just smash huge chunks of the lignin out of the way, make a giant gaping hole, and that will reveal the cellulose beneath. Once that's revealed, the fungus can extend its hyphaea in and begin to scoop out all of the cellulose, and that's what it actually eats. So even though it doesn't eat lignin, the capacity to chemically break it down is critically important for its ability to eat the cellulose of these plants. And as you'll see in a minute, it's also incredibly important for other reasons. Okay, so transplant that little sentence, delete the little footer header sentences, and I'm going on to the next paragraph right meow. Recall that the ability to break down lignin emerged sometime during the Carboniferous period, a brief stretch of time after lignin first evolved in plants. In that brief stretch of time, that brief window where lignin existed but nothing could break it down, entire forests of trees were growing and dying and growing and dying and going through the ages without rotting. That's what happens when nothing can chemically decompose something. That something that can't be decomposed tends to stay around for a while. Entire forests didn't decay, they didn't rot, and so they were preserved and fossilized. And this makes the Carboniferous period actually really rich in paleontological and evolutionary data. Here's the key takeaway that you should understand. If these ancient, unrottable forests, these jungles and boreal forests and rainforests and whatnot, if all of these unrottable forests of the Carboniferous had continued to grow and accumulate and soak up carbon, a huge chunk of the carbon currently cycling through the world's biosphere would instead be locked up in coal deposits, buried deep underground. Much of the coal that we mine today came from the Carboniferous period, so these would naturally be a lot larger if fungus and bacteria needed an extra 100 million years or whatever to evolve the ability to break down lignin. The ecological side effect was that by breaking down all of this lignin, and by making sure that you couldn't have just forests that would grow for millions of years and never rot, the fungi were able to return that carbon back to the carbon cycle, where it could then be absorbed by plants, and eaten by animals, and then eaten by fungus when those animals die, and reconstituted back into plants, and it just goes through this big carbon cycle. If the fungi did this millions of years later, if they evolved the ability to break down lignin millions of years after they did, or if they didn't evolve that capacity at all, the consequence would be a world without enough carbon to sustain the dense vegetation and the animal life that we see today. All of that carbon in all of these trees and animals and whatnot and other plants, instead of being available in the carbon cycle for all of this life, that carbon would just be sequestered hundreds of feet underground in coal. If this was the case, then the world as we know it would be a very, very different place. This is perhaps the most ancient, most fundamental way in which fungi have modified the world to make it more suitable for life. There are countless other ways that fungi modify the world, altering ecosystems and engaging in symbiotic behavior that promotes life where it would otherwise struggle. These symbiotic relationships between fungi and other living things play a defining role in fungal ecology, which I'll be exploring in the next episode. For example, I largely ignored mycorrhizal fungi in this episode, even though mycorrhizal fungi have a really unique and cool method for acquiring food, for getting their nutrition. However, mycorrhizal fungi engage in one of the most important symbiotic relationships in the world, so I figured it would be better to save them for the next episode on the symbiotic relationships fungi have evolved and their ecological ramifications. Anyway, if you're enjoying learning about the world of fungus, then get ready for stuff to get really crazy, because when it comes to symbiotically interacting with other organisms, nothing else on the planet is as diverse or as widespread or as important as the fungus. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.